thank you everyone for coming here. So today I'm going to give some update on our JFF grant, and thank you for the support. Yes, we do have Dr. Kapu, and she was on the team, but then she left early this year to Mars. The good news is, one of her colleagues, actually Dr. Martin, he continues to support us with this, with the related research on center study. So let's start why we need protein. Actually, with the growing population, you see the prediction of the population continue to grow, and then we need more protein to feed the future. So there's a prediction, for the demand of both animal protein and plant protein. And then as for the plant protein, particularly the majority of this plant protein available on the market are the wheat gluten and soy protein. So they two actually account for over 97% of total protein. But then, with the gluten allergy and also with like the GMO soy, and the industrial and consumer, they are looking for some additional source. Can we get some extra, better plant protein instead of with gluten and soy protein? That's why we are looking for some other plant protein. And then there are some advantage of utilizing plant-based protein compared with animal protein. So one of the major reasons is the sustainability. So like the conclusion is the production of plant protein is more sustainable compared with the production and the consumption of animal protein and animal food. Because the production of animal protein, like beef, like beef chicken, like pork, they took more land and also more water, and also like have more greenhouse gas emission. And also you need like more plant protein to produce animal protein. The data shows like it could take up to 10 pounds of plant protein to grow like 1 pound of animal protein. So the overall process is not sustainable. And also there are concerns about animal welfare or some like high cholesterol in those red meat or other concerns. That's why we need plant protein. So as for the plant protein, there are a lot of things to research. So basically like this graph shows like the whole a lot to like work in the field of plant protein or food protein research. So basically it's a study of this like chemistry and structure and the function and application relation. Because different plant protein, they may have different functionality. And then for that application of functionality, we know the nutrition is important, but then once they meet the nutritional requirement, we will talk to their functionality. Can they really be applicable? They deliver some texture, their function in the food. So for example, if we are going to make a beverage with plant protein, we will prepare that protein with some good solubility. And then if we are going to make a dressing with some plant protein, we need like to have good water holding or the oil holding or the emulsification. So there's different examples. And then the protein sources, where they are from corn, from lentil, they may have very different functionality. And other process, extraction, drying, post-processing, extrusion, texturization, all those will alter their chemistry and functionality. And also like the formulation of the food. So that's a whole, just a lot of things to research on. So let's move to our goal. Like my research goal is to like for long term, we want to generate advanced knowledge and science to develop a superior meat, dairy, and egg product with plant-based protein. And then we have like multiple objectives to achieve this goal. So for this seed grant back in 2019, the objective was to screen and identify plant protein with the most functional properties that could be used as an animal protein alternative. And then with that, we developed different additional objectives to the like structure formulation, and then their modification, and then their application, and also even like collaboration with industry. So I focused on this objective for this update, but we did get some extra grants to continue work on those additional objectives. So back to the plant protein, there are actually a lot of options. So obviously we do have different sources for the plant protein, from cereal, from pulses, oil seeds, leaves, vegetables. There are a lot of options. We look at the USD, like the food database, there are a lot of options. So then based on the availability, we pick up like some for our screening. So we look at the protein from pea, from lentil, from cowpea, pigeon pea, mung bean, oats, canola, hemp, quinoa, and also American leaf. That's not very common, but some people they are like that. and algae and even pumpkin seed. And I do have like the abbreviation for different proteins. And then for this study, we use pork protein as a control, as a pork malfibroprotein extract and comparison. And also use commercial soy, gluten, and pea, and also add for a benchmark and comparison purpose. So, we got the raw material at the meal. It's like we got starch, fiber, fat, everything there. So we take the protein out. So for the plant protein, the extraction, mostly it's just they have some unique solubility profile. So most of them are some like lobular proteins. So they are dissolvable at higher pH, and they can precipitate and separate at their isoelectric points. So based on this principle, we kind of extract those protein isolates or we concentrate from that like original meals or flowers. So that's a typical process. We take the protein out and then we freeze dry. We got a powder for the further analysis. And then for the muscle protein, they would have different whether it's solubility or the property compared to the plant protein. So we use a completely different method for the extraction of like pork protein, but also we work with beef, chicken, and fish muscles. We use a different method. We got that powder, like the malfibrate protein powder for comparison. So here's some results on the protein properties. We look at those are the sources, and then we look at the original protein content in that meal or flour. And then this is like the extract, the protein content in the extract. So you're like for the protein within like 70 to 80, we consider as a protein concentrate. And then from 80 to 90, we consider as a protein isolate. So it looks like most of those extracts, they have pretty good protein content. And also the protein yield. So here's the yield based on the flour, like original weight. And then this is a protein extraction based on like a total protein. So I highlight some of those like promising extraction because we took like 50, 60%, or even 80% of those protein out with our extraction. We consider that like more promising. And then with others, like the extraction is low, but we could possibly like optimize the process to take more protein. But for that seed grant, we just like not spend a lot of time on the optimization of the rest one, but we got some promising ones. And then we compare like the solubility. So let's show different proteins. Those are all the abbreviations we defined earlier. It's hard to memorize, but I will explain some. So the mark, that's a benchmark we use for pork. So the pork protein solubility. So we can tell like a lot of plant protein, they actually have a better solubility than those muscle proteins. And there are a few exceptions. Some are low, like gluten that is low, and also like the krill protein has a lower solubility than that one. Than pork. And then for the emulsification, so the emulsion is oil mix oil with water, and then with the emulsifier, they make the emulsion. Okay, that's so like the protein, they can be a very good natural emulsifier to make a homogeneous mixture between oil and water to measure the emulsion property of different protein. We can see again that the pork mark, so the pork mark for most of the plant protein, they have pretty good, like the emulsification properties comparable with pork. And then some are not that good. For example, like the commercial gluten, commercial soy, they are lower, and also for like the algae protein, like zero emulsification property. And then stability, they have a similar trend as what we found for the capacity. And also gel, because some food we like make a very nice gels with those proteins. So we measure the least gelation concentration. So that should actually lower number, the better. So like the number here, like 16, that means like 16% of that slurry to make a gel. So then the lower values mean like we require less protein to make a thinner gel. So the lower value, the better. So the pork, so 12% to make that gel, and then with other protein, a lot of plant protein, they actually better gel property than the pork. Then we have some like worse, like the commercial pea protein, commercial soy. Even like the lamb pea protein, they are not as good as pork. But then for some like the algae proteins and even for krill protein, they can make it like gel with 8% or 5% protein slurry. And also the water holding capacity, those are also very important. You can make like a patty from like plant protein. People always complain, like the ground burger is not as juicy as real beef, because they have very low, like the oil holding or the water holding capacity. That's why we want to get some better plant protein with water and oil holding. So you see the pork at this range, and then some protein, like a lentil, is actually very good water holding. And also some are comparable with this pork, but then some are lower. You have like the oat proteins or yellow lentil, and commercial soy, they have lower water holding capacity. And then for the oil binding, so as I mentioned, it's as important as water binding. So for the pork, it's 2 grams, and they got some protein, they actually double oil binding than pork, and some are lower. There are some examples, some like examples. And then we can make a table to rank those proteins based on their functionality. Again, you cannot tell like which is the best one because different applications they require different functionality. That's why we rank based on each individual functional property. We found some more among the top, those are the better ones. So we kind of conclude, like the pigeon pea, chickpea, lentil, cowpea, and algae, and also pea and quinoa, they have very good, desirable functionality for some further study. And also we compare with pork, we actually extract those animal protein from fish, from chicken, beef, and even compare with milk, the casein, or the whey protein with the egg, whole eggs, or egg white, egg yolk to get some mark as a functionality. So hopefully they provide some information as we are going to develop an alternative to those real animal food. So then we also work on some sensory studies because this was done early in 2020 with Dr. Cotton because of the COVID. We really like to sit together do the sensory. For that time, we actually prepare all those samples and we deliver to our panel's home because they are all in the hand, so they have to make it easier. And then we deliver in the morning and we meet in like the noon or the afternoon, and then we just meet online to score those attributes. So those are actually based on the P protein. We have a control P protein and with different like the modification of the P protein. And then we give like a vinny flavor, starting grain, see like with those modifications, we get like some kind of different sensory attributes compared to control. And then, after with that, screening, we got some nice proteins. We did further research. And once like we compare the post proteins, we have a very systematic study of different post proteins based on that study, the previous GFS study. And then we actually published in LWT earlier this year to run more information after this paper. And then we did another study on the drying because from quinoa is good, and also we found like the processing could actually make a different functionality. So we start like different drying methods, how they affect functionality of quinoa protein. So in that we do like spring drying or like drum drying, and then research like freeze drying. So we compare like freeze drying and spring drying, and also the vacuum drying, how they alter the functionality. We publish it in food chemistry. Also, we start the modification because natural proteins, they are what they are, naturally genetically controlled as a property. But if we want to improve some of the problem solubility or the oil binding, we can do some like modification with some biochemical or physical modification to improve functionality. So this study we use acylation and also the conjugation to enhance the functional property of the P protein we published earlier in the hydrocolloids. And then next one, the application, we got those good proteins can use it. The one study we did is we work with Dr. Chow and Dr. Quinn from animal science, and we draw like a little bit of our protein to the real meat, like as a functional extender. And we found actually with the addition of our modified protein, we got much better, like the like a lower cooking loss. Then we got better yield after cooking, and also got like softer texture. With some people, they can feel like the original meat too hard. With actually with like 2% of our modified protein, we got much tender and soft patches. So there are some advantages with this modified protein. And also, I was invited to provide like a perspective paper to the Zero Food work back in 2020 on my perspective on using the plant-based protein or the plant-based meat. So we published this one in 2020. And also in order to promote our own research and also to get more like knowledge sharing among the community. So I actually organized a special issues focus on the functionality and the application of plant proteins and also like to be worldwide for submission and peer review. So hopefully by next year, we're going to have some good paper publishing this issue. So then we are like considered pretty successful on securing some additional grants based on the GFS. So once, in 2020, with some data available, and then we prepare a proposal to the USD Post-Crop Health Initiative, and then we focus on the post-protein, like the pick up the lentil, pea, and chickpea. That's just some modification and application of post-protein. And also with that information, we work with pulse, and we feel, okay, actually the post-protein, they can be used as a bakery because it got like a complementary amino acid profile to the cereal. That's going to add some pot flour to the cereal flour to make some bread or some other bakery. So we focus on the rheology and the baking and sensory of this post-fortified wheat flour. So actually, in 2020, they received like 85 submissions, and they found 15 projects. And then we were like 2 of the 15 projects. So it's really fortunate at that time. And then we also submit a few other proposals, but not funded. So we submit to the Good Food Institute to focus on their meat quality and functionality. It's declined. And also we work on crop protein with Dr. Lavi and Dr. Chow to USDA. It's again declined. And then we submit to US Farming Board on soy protein. It's declined. And then we submit our on duck wheat to the Good Food Institute. It's declined. But we are keep trying to get more grants. It's normal. So we did have a patent, based on the USD grant and also some GFS support. We got this grant on the modified plant protein with enhanced functionality and application. So then we work with our IP, the Innovation Office. Sorry, our IP, I think, called some innovation partners or something. Yeah, the IP office. With Sarah and Chris, we work with different companies on those meetings and rooms and sample preparation. We're still working on this, just keep, hopefully we can apply our technology to their industrial system. And also we build some multiple collaborations after this grant, with Dr. Kaggle from sensory and Dr. Martin from sensory, Dr. Alavi on the execution, and also Dr. Kelly on the mailing. And also you see that our goal is going to, partially replace animal proteins. We need some animal experts. We work with Dr. Lee on the dairy and on milk proteins, with Dr. Chow on the meat protein, and then with Dr. Queen on the animal and sensory parts. And then we do have some workforce training. So once, I think a couple of my students are actually in this room. So Blake and Junjia, they might see some of our results in my class. I teach green protein chemistry and technology. So some of this data actually part of my lecture information. And then we have 3 key participants. So Dr. Yanting Xin, she submitted her dissertation this month. So she's going to graduate this year. So actually with her training on the protein, she's going to continue working on the protein at ADM. So that will provide a training for the industry. And then Dr. Hong, so she joined early this year at a postdoc to work on this protein project, got her PhD from University of Tennessee, and then work on protein. And also we have undergraduate setting, so she's actually helping our students with some sample preparation and some simple tests. And then we do have other participants, they have some, actually help with different part of this protein project. And then we do have some dissemination of those knowledge. So once dissertation submission, and also we have 5 papers so far, and also a few more are either under preparation or in review for the plant protein. And we do present our results during the last 2 to 3 years at FT and ACC and AOCS to share with different worldwide. So that's all. I got precious support from the CIV grant. With that one, we can secure the additional support from USDA and also my student postdoc, Wilton Starr. Thank you for being here.